Hello, my name is Dr. Roderick L. Rowe. Today I will talk about chemical evaluations. Methods for evaluating these chemicals or these disinfectants can include one of these three methods. We can use the phenol coefficient method, we can use the use dilution test, or we can use the in-use test. Any of these three can be used to measure the effectiveness of a disinfectant. The first evaluation procedure we will discuss is the phenol coefficient test. This test compares different dilutions of two disinfectants against two bacteria, one being Staphylococcus aureus and the other one being Salmonella typhi. You're going to take phenol and compare it against, let's say, bleach. Those are two disinfectants. You're trying to see if bleach is stronger than phenol or is bleach weaker than phenol. You're going to take each disinfectant and do a dilution series. Then you're going to contaminate each dilution series with the bacteria in question. And you're trying to see which disinfectant kills better. If it kills better than phenol, the number that it will receive will be greater than 1. If not, it will be less than 1. So look at table 7.6 and you will see a few different disinfectants being compared to phenol. Look at Lysol. Lysol is a little bit stronger than phenol in reference to killing salmonella. Here is another example. Chloramine is a disinfectant that we discussed earlier, and it is composed of chlorine, which is a halogen, and ammonia. Collectively, those two chemicals will destroy bacteria. When it comes to killing Staphylococcus aureus, chloramine is 133 times more effective than phenol. In reference to salmonella, chloramine is 100 times stronger than phenol. The next evaluation procedure we will discuss is the use dilution test. With this test, we're going to dip several metal beads, or they can be plastic, into bacterial culture. This bacterial culture is going to contaminate the bead. After the bead has been soaked, the beads are taken out and allowed to air dry. You want the bacteria to be able to somewhat adhere to the bead. Standard bacteria that will be used are Pseudomonas aeruginosa, Salmonella enterica, and Staphylococcus aureus. The beads are placed into the test tube with the respective bacteria. The bead is allowed to be coated with that bacteria. Then after the bead is pulled out and dried, it will be placed into different dilutions of that disinfectant and allowed to remain there for 10 minutes to allow the disinfectant to kill the bacteria. Once the bead has been soaked in the disinfectant, you then remove it. You need to rinse the residual disinfectant off. You will then air dry it, followed by placing the bead in fresh, sterile broth. Now you will use a spectrophotometer to determine which test tube is clear versus which test tube is cloudy. Another term for cloudy is turbid or turbidity. So out of the different dilutions in this use dilution method, you want to see which dilution killed best. Since the use dilution test contains a very specific bacteria under very specific conditions, the results can be misleading. So another method called the in-use test is more efficient at letting us know which disinfectant is best to use. The swab is taken before and after. Before you use the disinfectant, you will swab the doorknob. You will put that into a test tube containing broth. Then you will clean the doorknob with a disinfectant. Then you'll take another cotton swab and swab the cleaned doorknob. You'll take that swab and put it in a different test tube with broth. You will allow both to grow, and then you're going to check the media for growth. You're going to look for turbidity using a spectrophotometer. This is a more accurate way to determine if the disinfectant is good.